Hello, everyone. I am Nadia Selami, PacBio segment lead for neuroscience and biopharma applications, and I'll be the host and moderator of today's webinar. I want to welcome everyone to this webinar with today's topic focused on the use of HiFi sequencing to advance our understanding of neurological diseases such as Parkinson's. I'm joined today by our wonderful speakers. I've got Nicole Ellis-Avadia, the Associate Director for the Human Genomics segment, giving an introduction, and Dr. Mina Wrighton, the Van Geest Professor of Neuroscience at the University of Cambridge and Director of the Dementia Research Institute in Cambridge, who will present on her research after the brief intro. We have a lot of great material to cover today. Both the introduction by Nicole and Dr. Wrighton's presentation are pre-recorded, but will be followed by a live Q&A. You're welcome to submit your questions at any point during the webinar by typing them in the area provided on your attendee control panel, and we'll get to them either in the chat or at the end. We've also uploaded complementary literature to your control panel, so please feel free to download that and take it with you. We'll be recording this webinar and making it available for download in early January. Please keep an eye out for a follow-up email in the new year with a link to the recording. Immediately following this webinar, you'll receive a brief questionnaire. Please do take the time to fill it out as it helps us understand your needs and plan for future webinars. You can stay up to date on upcoming webinars and events by visiting and bookmarking pacb.com/events. So with that, let's get started with a brief introduction and then Mina Wrighton's research talk before we have the live Q&A.
Hey, thank you, Nadia. Hi, everyone. Welcome to our webinar today. We're really happy to have you here for this really exciting discussion on how PacBio can impact your neuroscience research. The challenge in understanding neurodiseases, as most of you know, is that the complexities in the genetics make them really difficult to resolve. Over 60 inherited neurological diseases are caused by repeat expansions, which are very difficult to detect with some of the traditional technologies that are out there. And also the disease onset and the severity can be linked to repeat lengths, which is, again, difficult for most of the traditional methods to be able to measure. And the accurate measurement of that is really critical for diagnostics, prognosis, and then further therapeutic development. So as we all recognize, these are very complex diseases. So here, the slide is meant to just really illustrate the power that long-read sequencing can bring into resolving these complex diseases. So on the left side, you see an example of a puzzle, which you've got a lot of pieces that are there, but you also have some missing pieces. So you really can't see the full picture, which is of a rhinoceros. And this is what happens with short-read sequencing. You really are missing a lot of pieces that just don't give you the complete picture. On the right side, you can see the impact that long-read sequencing has. You're able to solve this puzzle and see a complete picture because you're looking at bigger pieces of the genome and you're finding more complex features. This is also true on the RNA side that helps you really understand what's going on with your neurodisease. So this is a look at the timeline of really studying a lot of these neurodiseases over time. So you can see for a long period of time, there wasn't a lot of progress in really understanding a lot of these repeat expansion diseases. As we look and see in 2010, this is the start of the NGS era. So when next-gen sequencing was introduced, you really started to see an acceleration. But really, the exponential growth came in as soon as we started with the long-read sequencing era, which is where PacBio comes in. So you can see that 25 of these disorders are caused by repeat expansions, and those have been discovered in the last 10 years. So just a really significant impact and increase on the area of neurodisease research. Here's an example, just a few different publications that we have in different areas of neurodegenerative diseases that have been characterized and published on using PacBio long-read technology. So you can see all of these here, and feel free to, at the end, go take a look, read some of these papers, but you can see that we've had demonstrated success across bipolar disorder, schizophrenia, Parkinson's disease, which we're going to talk about in our customer presentation today, and many, many other repeat expansion neurodiseases. Some of the key advantages of HiFi reads for neuroscience are long reads, so we can capture up to 25 kb and find areas of the genome that might be missing using short reads. We have uniform coverage, and this better data means a better understanding of underlying genetic events in difficult to sequence areas that might be missed by other technologies. Also, you can have great confidence in your calls that will allow you to get to answers faster with our 99.9% accuracy or Q30 that ensures confidence in variant calling. We also have comprehensive data, so you can see the easy stuff without missing the hard to find. So again, you can get everything from the small variants that you can get with other technologies at a very high accuracy, combined with complex structural arrangements, repeat expansions, and then even a multi-omic approach that we provide. So I just want to point out, we actually just expanded our instrument platform. We now have 2 options for long-read sequencing. The first is our Revio, which has been out for a couple of years now. That is more for high throughput labs, but we now also have the Vega, which is a benchtop long-read sequencer, which allows you to do smaller experiments at a very affordable price point. So both systems give you the same level of complex data analysis, including repeat expansions, structural variants, as well as RNA isoforms, and then epigenetics with looking at methylation so that you can get a complete multi-omic picture. We also offer a suite of reagents across our instrument portfolio that help you achieve these goals. So again, we just recently launched the Vega, which is our benchtop sequencer. Here you can see some of the specs. You could do 1 SMRT Cell per run. You get 60 gigs per run. It's priced very affordably, both at a system level and a per run level, and allows you to get all of the same things that you can with the larger Revio system. On the Revio system, you can see on the right, you can run up to 4 SMRT Cells per run, 480 gigs per run, and it's a little higher price point, although we just lowered the price of this product as well, but it gives you the higher throughput that you might need in your lab. So no matter what your needs are, whether you're running small experiments on your benchtop, or if you are working in a larger core or other type of lab where you have high throughput, we have what you need to get long-read sequencing done. And then I also wanted to mention, so we are bringing the affordable genome into the picture as well. So our new Spark chemistry, which we launched, allows for a lower DNA input, so you can run more samples, increased output that help also reduce costs. So we're bringing the genome down to about $500 per genome. And then we also introduced a new methylation approach compatible with the FiberSeq assay, which again brings in the multi-omic aspect for you if you're looking across a bunch of different modalities with your disease. And this is available for pre-order now with shipments by the end of 2024. And as I mentioned the last slide, the price of the Revio has also come down, making the high throughput affordable as well. So thank you so much for your time.
So thanks very much for inviting me to speak to you today about long-read RNA sequencing of synuclein in Parkinson's disease and how it's provided us insights into the disease process and potential ways of treating Parkinson's disease. So I've entitled this The Splice of Life because I think really this technology has made a massive difference to our understanding of transcripts. My name is Professor Mina Wrighton. I'm Director of the Dementia Research Institute in Cambridge, and I'm Consultant Clinical Genetics at Addenbrooke's Hospital, again in Cambridge. So Parkinson's disease is actually quite an old disease, first recognized by James Parkin in the 1800s. And it's actually a disease that we have known about for a long time, but our understanding of the symptoms that actually make up Parkinson's disease has been growing. So in a way, it's an old disease with new symptoms being recognized, a wider recognition of the involvement of Parkinson's disease in affecting cognition, mental health disorders such as depression or apathy, as well as autonomic components of the disease process too. It's actually a disease which is also growing in prevalence, so it's not just because of aging that the number of individuals with Parkinson's disease is rising. There's also a lack of understanding about exactly why, but the fact remains is that the proportion of individuals globally with Parkinson's disease is increasing. So this is an important disease which at the moment is untreated, and we have no treatments for this condition at the moment that actually affect the disease course, making it an area that we really want to understand further. It is a place where genomics has made a massive difference to our understanding of the disease architecture. Much of this progress has been made with short read sequencing. So the development of next generation sequencing has meant that we now actually sequence more DNA than we ever did in an individual patient. We sequence more people than we ever did. So here I'm just highlighting the Global Parkinson's Genetics Program Initiative, which is actually sequencing individuals with Parkinson's disease worldwide and incorporates over 200 different cohorts of patients. Together, the higher coverage and the higher amount of data we generate per person and the larger number of people that we've been sequencing has meant that we've identified more disease loci for this condition than we ever previously knew, and are beginning to get an understanding of the architecture of that disease. Like many other complex diseases, Parkinson's disease has in common the fact that we have both very rare variants having high effect sizes, so the traditional familial forms of the disease, together with actually more common variants, often in the same genes, that actually give rise to lower amounts of risks but collectively increase risk of disease. And so this progress has been incredible, but at the moment it hasn't actually transformed treatment. And I think we have to understand why that is. Part of that is actually that genomic medicine depends on having an accurate map of transcripts, not just genes. And we can actually think of our understanding of the transcriptome of the human brain in particular a bit like maps of any other kind, so maps of the world. So if we look at the map from 500 BC of the world at the time, or our understanding of the world at the time, you can see that actually it's very, very accurate with regard to some parts of the world. So if we look at the Mediterranean, actually that map is near perfect for somewhere like Italy. But on the other hand, we're completely missing North America. We're completely missing Australasia. There are other parts of the world that are just utterly missing from this map. And we can think of the map that we've been developing of the human transcriptome a bit like that, where we're very accurate in some places and very inaccurate in others. Now, of course, the accuracy of that map has tended to increase over time. So here, what I'm showing you is the period of time between 2003 and 2024, where what we can see is that as sequencing technologies have improved, length of reads in particular have improved, and the quality of that sequencing data has improved, we identify more genes in the human genome, but we identify another thing that's really important. We identify many more transcripts. And that is really key. So it's fine if a map isn't perfect everywhere, but I guess the question is the map that we have of the human transcriptome good enough for human brain? And that's a question that my lab has been interested in for some time. And we've examined the quality of that map in human brain using short read RNA sequencing data in the first instance. And the first thing we did was a project that actually dates from quite some time ago now, back in 2020, which just used short read RNA sequencing data. And instead of mapping it to the known transcriptome build of the time, we simply mapped it to the genome and quantified expression across exons, introns, intergenic regions, and asked ourselves whether there was a more expression or more transcribed regions than we expected there to be. And we quantified that and quantified the length of those unannotated transcribed regions. What we found is that if we focused on intergenic regions or intronic regions, regions of the genome which should not be heavily transcribed, we found that in actual fact there were more of these regions transcribed than we would have expected, particularly in human brain tissues. So just to focus on that, you can see that on the x-axis of this first graph, we've got all the human tissues that were analyzed in the genome tissue expression database from the broad, and on the y-axis, we've got the amount in length of megabases of the novel expressed transcription from the intergenic regions. And we can see that all these yellow tissues, which are actually all derived from human brain, are all stacked at the top. And so really, there's actually regions of the genome or the transcriptome, particularly in human brain, which have just not been annotated and which have been relatively unknown. In this project, we also asked ourselves whether those regions, those novel transcribed regions, related to genes which were particularly expressed in one cell type or another cell type of the human brain. If I'm honest, at the time we were doing this project, I actually imagined that the cell type that we knew least about might be excitatory neurons. But in actual fact, what we found was that the novel transcribed regions we identified, if we tried to connect them to known genes using exon-exon junction reads, we found that those reads actually connected to genes which are often highly expressed in oligodendrocytes. And in retrospect, you can see why that's the case. Oligodendrocytes are a very difficult cell type to culture. They're a very difficult cell type to isolate on their own and to purify. And actually, that's one of the limitations of current sequencing technologies where we've used bulk RNA sequencing and we've relied on the frequency of a transcript expression and the ease with which we can model that cell type to be able to understand what the actual transcriptome of a particular cell type is. We also began to recognize that some regions of the transcriptome were better than others, and because of actually the problem of repetitive regions in the genome. And in a way, that's something which has been very well recognized for sequencing of DNA, where we've known for a very long time that genes like GBA1, for example, which have pseudogenes, genes which are almost identical but lack function, are actually very, very similar to the main gene. And it means that when you're using short read sequencing, so short read DNA sequencing, these regions will actually map with equal probability to both genes, the parent gene and the pseudogene, and it's very, very difficult to then identify whether a mutation has arisen in one or the other. And while that's been recognized as a major problem for sequencing of DNA, it's been less recognized that it's also going to impact on the quality of the transcriptome build and our knowledge of the transcripts generated from those loci. So, addressing this question, is the map good enough for the genes we care about most? Now, as I've said to you, I think that there is a problem with the quality of the transcriptome build for human brain, but maybe that doesn't matter if the specific genes we're interested in aren't the ones that are being affected. But in actual fact, our initial analyses, for example, this work in 2020, showed us that, in fact, many of the genes which were poorly annotated were genes which are already known to contribute to human disease. So these are genes which are within the OMIM database. So that's the database of genes and phenotypes that are inherited in man and which cause familial disorders. And we also found that in actual fact, particularly for my lab, who've been interested in Parkinson's disease for some time, we found evidence of novel exons of a gene synuclein, SNCA, the major constituent of the protein which is deposited in cells in the human brain in Parkinson's disease and which is thought to be key to the development of the disorder. We also identified in further work that actually we were likely to be also missing transcripts of the gene ApoE, one of the major genes which is a risk loci for Alzheimer's disease and is probably the single most important risk locus for that disorder. And we found that in actual fact, probable novel transcripts which retained a particular intron of APO3 were not only likely to be real, but also correlated with the levels of pathology in brains from individuals with Alzheimer's disease, the so-called BRAC staging, and that could be involved in the disease process. So again, focus on this question of, is our map good enough for the genes we care about most? Since actually those initial analyses were done with short-read RNA sequencing, we've gone on to use targeted long-read RNA sequencing, particularly with the PacBio platform, which actually was very effective in analyzing GBA, a key gene causing both a rare disease, Gaucher's disease, but also one of the major risk factors for Parkinson's disease. This gene has a pseudogene, GBA pseudogene 1, and this gene is actually 96% identical with GBA1. So this was a perfect use case to try and understand what targeted long-read RNA sequencing would provide us with, which short-read sequencing had not. This paper is now published, but actually what we did was analyze both GBA1 and GBA pseudogene 1 in brain and in iPSC-derived human neurons, astrocytes, and microglia. We used hybridization probes to capture GBA1 and GBA pseudogene 1 cDNA and sequence that at high depth using the PacBio platform, the Iso-Seq platform. This allowed us to identify novel protein coding structures of both GBA1 and demonstrate the GBA pseudogene 1 also generated potential novel coding transcripts, which in actual fact we later found out were in fact translated in the human brain. So this work, as I say, is out. What I was going to talk to you about today was another example of a gene that we've been very interested in, and that is synuclein, SNCA. SNCA has a very long history in Parkinson's disease. It's known to be the major constituents of Lewy bodies. So this is the pathological material that is deposited in neurons of the substantia nigra initially, but also more in other areas of the human brain, and which are key to the diagnosis, the pathological diagnosis of Parkinson's disease, but have also been key to our understanding of the pathogenesis of this disorder. As I said, alpha-synuclein was found in 1997 to be the major constituent of Lewy bodies, and as the years went by, we began to understand that actually mutations in synuclein, rare mutations, and multiplications of this gene were a cause of familial Parkinson's disease, particularly with major psychiatric problems. As the years went by into the 2000s and 2010s, there have been genome-wide association studies which have again emphasized the importance of this gene in the pathogenesis of Parkinson's disease. We found that in actual fact, common variants that regulate synuclein's expression and increase its expression are also risk loci for Parkinson's disease in sporadic patients. So again, focusing on Parkinson's disease and focusing on maybe the most important cell type for that disorder, the dopaminergic neurons, which are located in the substantia nigra, and which, when they die, is thought to be secondary to the deposition of Lewy bodies, you get the characteristic movement disorders of this condition, which include a tremor, bradykinesia, and rigidity. And so what we decided to do was try and really understand how synuclein is transcribed, what transcripts are actually made. Is it really just one single major transcript that's protein coding and can aggregate into these Lewy bodies, or is it more than that? So to do that work, we wanted to analyze a cell type that would be most relevant to the disease process. And for that, we actually used iPSC-derived dopaminergic neurons. These were generated in Sonia Gandhi's lab at the Crick Institute and have previously been characterized in work from her laboratory as a model of Parkinson's disease. We analyzed both normal dopaminergic neurons and dopaminergic neurons carrying pathogenic mutations in synuclein. So that's the A53T mutation and also the triplication of synuclein. So in order to study these dopaminergic neurons in great detail, we turned back to the protocol we'd previously used to study GBA1 and GBA pseudogene 1. This involved the purification of our cDNAs of interest and the selection of those cDNAs with IDT lockdown probes, a protocol that actually was originally developed by PacBio and which inspired the GBA1 work and which we later reused again for studying synuclein. We then went on to do targeted long-read RNA sequencing of synuclein using the PacBio Iso-Seq platform. And actually, this was worked very successfully in our hands. So, when we did this work, actually, we didn't necessarily expect to find as much as we found. And I guess I'm just going to focus your minds again on the depth of the sequencing and the quality of the sequencing data we obtained. So using the protocol, which we've published now on Protocols.io, and I think is also available through PacBio's website, we managed to generate really quite large quantities of high quality, full-length reads on all our samples, which were over 40 samples, I think 48 samples in total. So from these reads, we actually only recognized transcripts which were detected in all our samples and with a minimum usage of 0.3%. This equated to 300 full-length supporting reads per sample. So these are very high confidence transcripts. So you might argue, look, if you're actually using such a stringent cutoff to recognize a novel transcript, can you really find anything new at all? And so it really was surprising that despite being so stringent, we really did find a lot that's new. And in actual fact, what surprised us is how little of what was known we found. So on this first graph, you can see that we've categorized transcripts according to whether they are known in annotation, whether they're complete match from the 3' end to the 5' end, whether they have novel 3' and 5' UTRs but yet have a known coding sequence, or whether they might even encode novel proteins and have a novel open reading frame, or whether they're non-coding novel transcripts. When you look at this graph, what's striking is that actually relatively few of the transcripts we detected are coding known, where they're a complete match. We found many transcripts with novel 3' and 5' UTRs, and most surprisingly, we found novel coding transcripts of synuclein. I think the other thing that's really important is the usage of these transcripts, and this is demonstrated on the other plot that I'm showing you here. And what you can see on the x-axis is transcript ranks. So we're simply ranking the transcripts in terms of their usage, and on the y-axis you can see the percentage usage. And that y-axis is pretty important. That y-axis does not come even close to 100%. It doesn't even go to 50%, which is indicating that we had no transcript in this pure cell type, or relatively pure cell type, 85% purity of dopaminergic neurons in culture. We had no transcript in this setting, which accounted for over 50% of the total transcription from the locus. This is surprising. I genuinely think it is surprising. Now you can see that actually the top 4 transcripts all have a known open reading frame, but novel 3' 5' UTRs. But the next transcript, the 5th most common transcript, is actually predicted to create a novel open reading frame. And it's only when you get to the 6th most commonly used transcript at less than 5% that you've actually got the complete match. So really, this is a gene which has been very well studied, is very well recognized. Its importance in disease makes it a high point for research, yet we actually knew relatively little about its transcription in the cell type which it has been most studied in. So what are the implications of this novel SNCA transcription for our understanding of the disease process? So I think there's 2 ways we can think about this. One of them is that SNCA protein causes or is thought to cause disease through its aggregation. So alpha-synuclein monomers forming oligomers and then protofibrils and ultimately fibrils. And it's this aggregation process which is considered to be what generates the toxicity in the cells and ultimately kills the neurons in the brain. So we could ask ourselves, are any of the open reading frames that we're seeing, the novel open reading frames, do they have different biochemical properties to the known isoforms of SNCA, and therefore might they have different aggregation properties? We can also ask ourselves about SNCA RNA targeting. So in actual fact, trials are underway that target SNCA mRNA, aiming to have antisense oligonucleotides that might bind to the 5' or 3' UTR end of the mRNA and trigger its degradation to reduce the total mRNA and to reduce the total protein, and so ameliorate the condition. So this is an area which is of great interest. Is there any implications to our identification of novel 3' and 5' UTRs in terms of how this targeting should work? So just to focus on the first part, the novel potential protein coding transcripts of SNCA. So here on this plot, you can see in the top part that we've plot out the structure of the main select transcript of SNCA. That's the most commonly recognized form of this gene, which encodes a 140 amino acid protein. And we can see that in actual fact below it are these novel PacBio called transcripts that are just numbered really, which actually are predicted to have new open reading frames. We've highlighted sections of that, and we've actually highlighted in blue and red how they differ, how these transcripts differ from the main select form of synuclein. Now, what's important is that these novel open reading frames, I mean, this is important from the point of view of detection and also follow-up of this work, is that actually some of these novel open reading frames would be predicted to generate not just shorter isoforms than the 140, but isoforms that have an entirely unique amino acid sequence within them and that differ in their predicted amino acid length. And that gives us the potential to try and identify whether in truth these exist. So this is actually what we did, and we did two different approaches to try and identify whether these novel transcripts of SNCA that we predicted have open reading frames could actually encode novel proteins. One approach we used was the cheapest approach, which was to develop some new software to try and understand whether actually the open reading frames we generated, they would be predicted to have novel amino acid sequences. Could we find those novel amino acid sequences in public mass spec data that might have already been generated from the human brain? And to do this, we utilized Metamorphosis, and we had to generate some new software, which we call transcript to protein. This is actually available now, but is also being developed as a tool and will be published soon. And this was actually remarkably successful. Using this approach, we could indeed see that at least two of the novel open reading frames that we predicted could be detected through public mass spec data. We also used another approach, which is actually more work intensive, but again, based on the fact that there are novel amino acid sequences here. And that is where we used human brain samples, both the substantia nigra, this key region which we know is very susceptible in Parkinson's disease, as well as the anterior cingulate cortex, which is also affected in the later stages of the disease. What we did was take samples from these brains, we generated tissue lysates, and using antibodies against synuclein and regions which we thought would be common across the different isoforms, we could pull down and purify synuclein protein and then apply mass spec here and look at the peak sizes. Using both these approaches, we identified that indeed we could see that some of these predicted open reading frames were indeed making novel protein in the human brain, and this was for two isoforms, one predicting an amino acid length of 113, another one 118, which were both shorter than the normal standard 140 amino acid structure of synuclein. We also went on to use a program CAMSOL that predicts solubility of the proteins. And indeed, we found that, in fact, all these predicted novel isoforms of synuclein, the 115 isoform as well as 113 and 118, would actually be expected to have lower solubility. This would actually, in fact, indicate that should they really be in the human brain, you might expect them to aggregate more rapidly. And this is work that we're now following up, particularly with Michele Vendresculo's lab in Cambridge. So what's the importance of understanding these transcript structures, maybe not for the open reading frames, but from the perspective of the UTRs and the implications that has for design of RNA targeting therapies? Well, this is another component which we explored. And in this area, what we did was look at the variability of the 3' UTRs. What we're showing here is that actually we detected quite a wide range of different and unique 3' UTRs. These differed in length quite considerably, and in actual fact, because we had such high depth targeted long-read sequencing, we could actually quantify the number of transcripts generated from the SNCA locus that had a specific UTR. And so we could actually consider what the relative importance of each of these UTRs is in generating synuclein protein. And this is what this figure shows. This work is publicly available on bioRxiv. So what we could do was actually design antisense oligonucleotides that would be across the entire UTR space. And we could ask ourselves, well, look, based on this, which ASOs rather, which would we expect to be most effective? So that ASO is likely to be in this ASO design, ASO1. An ASO which actually would be expected to cover most of the unique 3' UTRs, as opposed to the ASOs like ASO9, which would only cover a subset of them. And so what you can see actually is that using this kind of information, we should be able to design antisense oligonucleotides more effectively and potentially make this process more targetable if we understood what the real transcriptional landscape was. So what implications does that have for actually our understanding of how we might actually target in real life, and how did that work? So in order to assess this, we use the SNCA triplication model. So these are iPSC-derived dopaminergic neurons that carry extra copies of synuclein. Therefore, they actually have high levels of synuclein protein. This results in high levels of aggregation and in high levels of cell death. And that's what you can see on this top set of 3 images here, is the high levels of protein, high levels of aggregation, high levels of cell death that we see in synuclein triplication model, where you've got control ASO. So this is scrambled. When we actually used ASO1 with the same backbone and applied it to this model, you can see that in fact we reduced the SNCA protein levels quite considerably, and this was most efficient with ASO1. We reduced aggregation, and we actually reduced cell death. So what we could see is that actually understanding the transcript structure enabled us to create a more efficient ASO design, and that's really because we better understood what we were designing against. In any other setting, you wouldn't actually design without knowing the structure that you want to design against. And given the quality of the technology now available, there seems little reason for RNA targeting therapies not to consider doing a proper analysis of the RNAs which they're targeting. I think this also gives us scope to imagine that we can actually make ASO therapies more effective and safer, and with a reduced side effect profile. So I think using long read in this way will likely have a great influence on the development of RNA targeting therapeutics. So what did we learn from SNCA? I think we learned that there are novel protein coding isoforms of SNCA, and I can't emphasize enough how surprising this is, because actually this is an incredibly well-studied gene. I think this does have potential consequences for our understanding of how aggregation might occur. Remember, this is an amyloidogenic protein, and so even small quantities of something that could seed aggregation could potentially be very important, even if it only accounts for small percentages of the total protein in the cell. I think we also saw that there was very high variability in the 3', 5, and 5' UTR use, and that there was no actual dominant transcript in the cell type. And I think this is a really important lesson for cell biologists, because often they model the disease using a single transcript structure, a single open reading frame, but even not just a single open reading frame, a single 3' and 5' UTR. And so I think this should actually affect our understanding of what's the range of transcripts which we could need to target, what's the range of functions that the gene could have. Then finally, I think, and most directly, I think we can already see that novel transcript discovery has implications for RNA targeting therapies. I think it should enable us to produce better quality therapies more efficiently by understanding the landscapes of transcription that we're actually trying to target. So I think that leaves me to acknowledge the people who really provided the samples that mattered here, and that's the patients who donated both samples for the iPSC work and in other studies, the brain tissue. Also, members of my own lab, and here I'd like to highlight Emil Gustafsson, who's both driven the work on GBA1 and also driven this synuclein project in terms of the practicalities of doing the long read, but also the very hard task of analyzing that long read sequencing data and visualizing it. At UCL, I need to thank John Hardy and Henry Holden in particular, as well as Nick Wood, who've contributed to the development of these protocols and the samples and the availability of the equipment. Sonia Gandhi's lab, I think this project would not have been possible without her laboratory and their expertise in the modeling of Parkinson's disease. And in particular, I want to highlight the contribution of James Evans, a PhD student, well, now postdoc, who generated the iPSC models and was also involved in generating the long read sequencing data and its analysis together with Emil. As well, I think I need to thank members of UCL Institute of Child Health who generated the proteomics work, and Michele's lab in Cambridge, who have been very important in helping us understand the implications of novel protein coding transcripts in terms of aggregation. And finally, and not least, of course, is my funders, and particularly the Dementia Research Institute, Aligning Science Across Parkinson's Disease, and the Michael J. Fox Foundation, who've really been key to enabling this work and the resources to make it happen. So thank you very much for your time.
Hello, everyone. Welcome back. Now we have some time for a Q&A session. Please remember you can submit your questions at any time in the question module, and we'll be answering them one at a time. There's been some questions already submitted through, so I'll go ahead and distribute those, read those again. Dr. Wright, and thank you so much for your talk. Someone here was clearly very inspired and is asking, how can someone get involved in the research you've outlined?
If you want to get involved in the research I've outlined directly with me, then please do send me an email. You can find me from my UCL email or my Cambridge email. I can easily pop that in the chat. I think if you're talking about getting involved in general, I just think that this is an incredibly exciting area. I think RNA biology hasn't really been possible to study well until there's been availability of high quality long read sequencing. So I think this is, I'm sure I'm not the only one who has this level of excitement about this area. So I'm not sure where in the world you are and whether Cambridge University in the UK is applicable to you, but I definitely would say I think this is an area of growing importance with growing relevance for therapeutics.
Great, thank you. And speaking about transcriptions, Nicole, there is a question whether it is possible to obtain the sequences of promoters or transcription factor binding regions.
Yes, so theoretically you can do these on our long read platform, but this is where short read might really excel and help you get a little bit better results. So we do have our Onzo platform, which is short read, that you could consider for that. So it can be done, but short read is usually more sensitive for this application.
Great, thank you. Nicole, you also mentioned the Spark chemistry. Will that be available for Sequel II machines?
So we don't currently plan to have the Spark chemistry on the Sequel II right now because we just launched it in November. It's only available on our Revio platform. We are looking for expansion plans probably into the Vega space, but currently we don't have any plans to put it on the Sequel II.
Thank you. Dr. Wrighton, how important was it to perform the target capture? Would you have obtained the same results from standard Iso-Seq?
So I think actually in this case, I think it's very important. And I think it's very important for additional applications too. So I'm really glad someone's asked that question actually. So I think the reason why the targeted capture matches so much is because actually you then in fact have very high confidence in the transcript structures you're calling, because actually the depth of sequencing you're getting over the gene that you're interested in is key in fact. The other place which I think people are going to increasingly find where targeted capture matters, and which we also are preparing a manuscript for at the moment, is around using the long-read RNA sequencing for allele-specific or allelic biases in gene expression. And I think there again, the key point is that you need a lot of reads to have confidence about aligning to a specific allele. So I think it made a very big difference, actually.
Great, thank you. Follow-up question to that came through the Q&A. Is it necessary to isolate those RNAs from the nuclei, or can they be found outside the nucleus?
So actually, this was in fact these iPSC-derived dopaminergic neurons. We were actually isolating poly-A-selected RNA, which was in actual fact going to be primarily cytoplasmic, because we weren't selecting for nuclear RNA. We have been playing a little bit with long-read RNA sequencing from single nuclear RNA libraries. My gut instinct, and this comes not just from the long read experience, but from experience with short read RNA sequencing, is that there are challenges to interpreting nuclear RNA. I think there are also opportunities. I think you need to know why you're doing it. So I think, of course, if you're interested in splicing, the problem you've got using the nuclear RNA is that you're going to have pre-mRNA. Now there, the long reads really, really are going to matter to you a lot. because actually you aren't going to be able to get through the read without having the long reads because of these long introns. But then you've got partially processed RNA, so it will make the analysis more complicated for sure, and it does. But I think there are benefits to using both, and I think it's really about your application or your interest area.
Great, thank you. Someone asked if any epigenomics have been or technically can be analyzed from these long reads specific to the brain or neuronal cells.
I think this is going to be another really exciting area. I mean, if I'm honest with you, I think actually, so I mean, obviously we're interested, in fact, and this is coming from a project that is some of the data is in this paper, which is on the SNCA triplications. So, of course, if you have triplicated a CNV, you usually have not just got a simple copy number variant, you've also got, you might have actually not exactly quite the copies that you expect, plus almost certainly you're going to have epigenetic changes onto the additional copies. So we are in actual fact very curious about this and are sort of weighing up how much time to spend on this at the SNCA locus. Some of that will be at the DNA level, and I think that's important in order to map the DNA outputs to the RNA outputs. I think some of that is also quite important on the RNA modifications as well. And I think I'm not an expert necessarily in RNA methylations and RNA modifications, but of course that's an extra level of information which I think is going to become increasingly interesting. And for cancer, it already is there. There are already manuscripts in that field which I think are looking very exciting. Applications of this in neurodegeneration, I think, are going to come through as well.
Great, thank you. As a follow-up, if you want to see 6-methyl-C methylation, or you also mentioned 6-methyl-A methylation, 5-methyl-C and 6-methyl-A, like what do you have to do to see that on PacBio? Do you have to do bisulfite conversion or anything like that? I believe the 5-methyl-C, that just comes automatically with each DNA sequencing run, so you don't have to do the bisulfite for that. There's a question for Dr. Ryan. How do you think about the lack of a dominant transcript? Is each cell making a variety of transcripts, or would each cell have a dominant transcript and it's just different from cell to cell?
That's a really lovely question. I don't know who asked that. It's a beautiful question to ask, and it's one that I would really like to know the answer to. I think I've got some gut instinct that I think that I actually think that each cell is probably making a variety of transcripts and doesn't necessarily have a dominant, is my gut instinct. But I think it's probably going to be variable across genes. So I'm not sure there's a very simple answer to this right now. I think it'll be really interesting to find out. I think it'll do cell biologists' head in though, because it means you have to be much more careful about your experiments than you had to be before if you don't believe there's a dominant transcript. But I think that it's an incredibly good question. Yeah.
Thank you so much. Here's a question regarding the sequencing depth, a follow-up question to an earlier question. Can you speak a bit towards what depth you need with a targeted RNA sequencing in order to assemble transcripts?
I mean, to assemble transcripts, you don't need a depth to assemble transcripts. The beauty of this is that even, and we loved it, right, is even with GBA1 and GBA pseudogene 1, which are 96% identical, and with a short read RNA sequencing, we actually checked our own data on this. So we had short read RNA sequencing from an Illumina platform using frontal cortex samples. So very, very similar types of data. And we actually checked how many of our reads uniquely mapped. And these were not that short reads. These are 150 base pair short reads, right? So not like 50 base pairs, 75 base pairs, reasonably long. And given that an exon is 100 base pairs on average, you know, you should be mapping pretty well. We found that actually only 40% of the reads uniquely mapped, right? So you are actually having 60% of your reads ambiguous to the GBA and GBA1 locus. On the other hand, if you're using long read, I think it was less than 1% where we had any ambiguity about mapping. So I think the reality is that you do not need the depth for the assembly. That's the beauty of this. You don't need it for assembly, not in the way that you need it for short read. The depth really depends on how much variety of transcription you think there is. So we are seeing some genes where really there is a major transcript, and you've captured pretty much everything you really want to capture other than noise, with relatively low depth. If, on the other hand, you have a gene that has very many transcripts, all used about equally, then you're going to have to go to quite high depths to get to some of these transcript structures. So it's not about assembly in this case. It's about variety of transcription in the sample set that you've got.
Great. There's another question here regarding the pseudogenes. Is there a possible way to identify a pseudogene from a parent gene before sequencing in order to prevent spending time sequencing a gene we don't want to?
So that's a good question. So I think the thing is in terms of you mean in terms of for capture. This will depend on actually if you want to do targeted long read sequencing of a particular gene and not capture its pseudogene. So first of all, a lot of genes don't have pseudogenes, but I'm sure this person knows that. If they're unlucky enough to be studying a gene which does have a pseudogene, then the degree to which you're going to have a problem of duplicate capture actually depends on how similar those two genes are, and therefore how many of the IDT probes can you design that would be specific to your gene of interest. What I would say to you, though, is you might think you don't want to know about the pseudogene, but I'm increasingly, there's a lot of cautionary data out there to suggest to you you absolutely want to know about the pseudogene, because actually a lot of them, as in the case of GBA1, are turning out, GBA pseudogene 1 are turning out to be potentially protein coding. So I know you may not want to capture that pseudogene, but think carefully whether you really believe that before you start, and it will very much depend on the sequence similarities to how well you can capture one without capturing the other.
Great, thank you. You mentioned you spoke about the ASO that may be a possible treatment. So someone is asking if this mRNA treatment is developed, how will it be delivered to the target in humans?
Okay, so actually, ASOs are already given to human beings. So I'm a clinical geneticist, and in actual fact, ASOs have revolutionized in a way the treatment of SMA. So that's particularly a pediatric spinal muscular atrophy. It's a pediatric disorder that is really quite severe. It has multiple levels of severity, in fact. There is an ASO that's delivered to patients. It's intrathecal delivery to deliver to the neurons, and it's actually in order to it targets, in fact, the pseudogene. That's why I would say to that person who's worried about their pseudogene, you may want to know about the pseudogene. So it actually targets it's not a pseudogene anymore. It's SMA. So the disease is caused by mutations in SMN1, and in actual fact, the ASO targets SMN2. And SMN2, you can have the better SMN2 works, the more it compensates for lack of efficacy of SMN1. And so that's how the ASO works. There's also ASO treatment for muscle disorders, for Duchenne muscular dystrophy. I think that's delivered by injection into the muscle itself, but I could be wrong. ASOs are also being delivered to the liver for liver treatments, and those are already approved again. And ASO treatments are in trial for eye disorders at the moment. So, and new delivery systems are coming through. So I really think that I know that there are thoughts about ASO targeting for renal disorders now as well. But the classical has been intrathecal for brain disorders.
Great,
I
thank
hope
you
that
so much.
answers. This is one of these areas I find really exciting. So yeah, I could go on
Yes,
for it longer. But
all the new treatment modalities that are emerging.
yeah, exactly.
That's great. So you mentioned a number of different studies in your talk. Someone is asking what's the best way to gain more information on these studies discussed during your webinar?
I think the studies that are referenced, like the GBA1, we put everything on bioRxiv, so all the SNCA work is actually already on bioRxiv, including the protocols. So that's there. For GBA1 and GBA pseudogene1, that's now published in Science Advances. For the work on target to therapies, target to protein, that's code to be able to go from long-read RNA to looking in mass spec data is also in the public domain. And that manuscript, well, we should have had it out already, but I think in the next month we'll be putting a manuscript out on that. So this is, and if there's anything that anyone wants to contact me about specifically, we try and be quick to put our data in the public domain, actually. So I think, and if we haven't put something, just let us know. And if we can, we will pass it on to you.
Great. Actually, this is a question from me now. You mentioned the transcript to protein tool and that this might be available as well. So where could people find that?
I think at the moment that's got a GitHub link. I'm going to have to search out, but it's on GitHub. I know that. And maybe Nadia, if I can follow up with you afterwards so that you can distribute. But we're putting a manuscript out on it just to explain its usage and applicability as well.
Oh, great.
We're kind of excited about doing that, and we're really hoping that. Oh yes, someone's put it out already. Thank you very much. Someone's put it on chat. Because it's under somebody, David Murphy, who works with Emil and I, who's been working on that and optimizing it.
Oh, that's great. Thank you, David Murphy.
So someone's been very quick to kind of, I think it's, it's Emil Gustafsson again, who wrote a lot of the code together with David Murphy, who's also in my group and in UCL. So,
wonderful. I'm not sure if everyone can see it. I'll put it in the chat for everyone to see right now. Everyone.
Now we're going to be under pressure to release the manuscript now, Nadia. But that's a good thing. I'm not going to complain. We want it used.
Right. Great. Okay, so we're coming towards the end of our webinar. I would really like to thank Dr. Wrighton and the PacBio team for their time and for sharing your expertise and experience. Just a reminder for the attendees, immediately following this webinar, you'll receive a brief questionnaire. So please do take the time to fill it out. And thanks again for joining us today. I hope you'll join us again for a future webinar. And with that, take care and have a great day. Thank you.
Thank you.
Thanks very much. Thanks again.